we know that a gene is responsible for production of a polypeptide. Okay, that was what we had started with our understanding as the main basic point. Now, what we study gradually is that it is by the formation of RNA, there is an entire process known as transcription. It is by the process of that transcription an RNA is formed, and then later on that RNA information is decoded into polypeptide. Now, this was a very exciting field of discovery or invention for the scientists who were studying about the decoding of genetic code. Okay. It might not be exciting for you, but for those people who have keen interest in this entire gene and its expression, it was quite an exciting discovery, and it was of great importance. So, we come to a term known as genetic code, that there is a code according to which the decoding takes place. Now, what is decoding? Supposedly you are given a code, and inside that code there is something hidden. Again, I would give you an example of movies. You must have seen that there is a treasure hunt and they have a code and they keep on finding what is behind those codes. In the same way, we have the genetic code where a lot lies behind AUGC. Now, what are AUGC? AUGC are the biochemicals or the nucleotides which are found in RNA. Why I did say ATGC? Because DNA has been left behind. DNA has been used to get mRNA. That mRNA is having the introns and exons, they also have been spliced. Now, this mRNA that has been produced or that has been obtained has to be converted into proteins, or rather I would say polypeptides. Now, from your knowledge of biomolecules, what are polypeptides made up of? They are made up of amino acids, right? Amino acid chain is a polypeptide chain. The polypeptide is formed when many amino acids are bound to each other by peptide bonds, and that forms the polypeptide chain. This is what is to be recalled from your knowledge of biotechnology. Now, what we have to pay attention towards is how many amino acids are present. They are 20 in number. Now, some relation or some sort of interconnectivity has to be maintained between, or has to be devised between these amino acids and what lies in mRNA. What is mRNA? mRNA is nothing but alternate arrangement of these four nucleotides, AUGC, either it could be GG, AUUCC. We have seen that there are many possibilities of arrangement of these four different nucleotides. So, mRNA has the language of AUGC, and proteins would be formed from amino acid. So, some link has to be developed between these two. Now, many scientists started studying about it. Now, before we get to what they had studied, we have to see that 20 amino acids would be coded by these four nucleotides. So, one single nucleotide cannot code this. Alright, why? Because there would be only 4 amino acids. If a single nucleotide coded for one amino acid, that is not possible, because certainly there would be 4 probabilities only if we had one amino acid being coded by a single nucleotide, as there are 4 nucleotides or 4 biomolecules that we have. So, there is possibility of only 4, but we are seeking 20 amino acids. What I am saying, if you are not understanding, is that because mRNA has the language of AUGC, and we have to find the possibility of 20 amino acids within these 4 only, then only we can establish a link between the two. And need not to tell you, whatever is in the polypeptides, that has to be coded from here. Ultimately, it is the mRNA which is telling how many polypeptides, what kind of polypeptides are to be formed. That means all the information lies here, and this is nothing but this AUGC, that is all different arrangements. So, all the information lies in these 4 alphabets which represent the specific nucleotides. Now, supposedly we have to seek 20 amino acids. If one alphabet is coding for one amino acid, then there are only 4 possibilities, because then it would be 4 power 1 and the probabilities that we have would be equal to 4. Then there another possibility is that what if each amino acid is coded by a pair of nucleotides? Then what probability do we have? We have 4 and the pair is coding, then also we don't reach the desired number of 20. It stops at 16. Then there would be 16 probabilities out of these AUGC and we still don't have 20 amino acids which would be responsible for formation of polypeptides. Then there was a scientist known as George Gamow. He studied this and he told that what if it is a triplet? There are 3 nucleotides which are representing 1 amino acid, then we get 64 probabilities. And 64 very well takes in 20 probabilities. So, it would be a triplet. Instead of a single nucleotide or a double nucleotide, pair of nucleotide, we will have a triplet where there would be 64 probabilities of getting a triplet and out of these 4 nucleotide based sequences. Now, this was proposed and then altogether lot of studies started in this field. Once it was proposed, he was a physicist, George Gamow was a physicist. He proposed that it is better to use triplet as the base which would represent the amino acids, and it very well takes into account the 20 amino acids that we had. Now, people like Nirenberg, Severo Ochoa, Hargobind Khorana and not to forget Crick, they started working extensively using homopolymers of RNA, like supposedly they are having U. So, they will have U, A, or copolymers, and they were having double the number of nucleotides. They started using it. It is a complex process. Supposedly you reach higher classes of your understanding. In higher classes, your understanding would enable you how they had deciphered the genetic code. So, we had arrived, as we understood, at a point where a triplet was coding for amino acid, and these scientists, as I named you, they were studying extensively to find out which amino acid is coded by which sequence. Okay? So, that gave us genetic code. Now, when we study genetic code, we have to understand a term known as codon. What is codon? Codon is nothing but that triplet which coded for an amino acid. The triplet that contained 3 nucleotides. Okay? And there were 64 probabilities out of U, A, G, C. This is what a codon means, and this is what genetic code was all about, that you had a triplet which would be responsible for coding for an amino acid. Various studies led to the proposition where we have the various codes for various enzymes, and you have to keep in mind that Crick gave us a very simple Wobble hypothesis, that is a simple proposition where we get an idea how to name the enzymes. There are not enzymes, sorry, there are many amino acids, like we have lysine, we have glycine, we have serine, we have methionine. So, Wobble hypothesis says that because amino acid is coded by a triplet, supposedly A, C, C is the triplet, alright. So, if the amino acid is to be coded, the amino acid supposedly I am talking about tryptophan, just for an example, or we will take glycine. So, it has GG, okay, GG, C would code for glycine, then we have GG, A would code for same, GG, U would code for same, and GG, G would code for same. So, this Wobble hypothesis says that for single amino acid, it is usually the first 2 alphabets are similar and the third one is changing. Why I have told you about this Wobble hypothesis? So that when you are learning the name of amino acids as coded from the codon, it is easier for you. Just see that what are the 2 initial alphabets, and from that you can learn the name of amino acid associated and you can replace it according to Wobble hypothesis, and it would be easier for you to learn. This is all about genetic code that you have to understand. the next lesson that we have, it deals with the characteristics of the genetic code, okay, the salient features. For that, watch the next lesson.